Okay, today I want to give you a very quick video about RNA-seq and how we use this technique to answer questions about the biology of the phage, especially after we have annotated the genome. So I'd like for you to understand what kind of questions we can ask using RNA-seq, be able to describe the basics of the RNA-seq methods, and be able to ask your own questions about the phage genome that you're studying using the RNA-seq technique. So I wanted to start talking about the QUK genome because this is a phage on which we've already performed some RNA-seq experiments. And this is a pretty special genome. This phage was isolated at the University of Maine last year by phage genomics students. And at the time, it was a singleton phage. It now belongs to cluster AC with one other phage. And these two phages are really, really weird. Their genomes, in some ways, look similar to genomes we've seen before, but we have some questions about the biology of this phage that we're not able to answer simply by annotating the genome. So let me give you a quick tour of the genome. So this top layer of genes here all appear to be structural genes, and you should recognize the portal protein and tape measure proteins. In the second row, we see some more structural genes, the lysins and the holins. Then we see a stretch of genes here that we have no idea what they do. We see another stretch down here that includes some membrane proteins. Finally, we've come across a gene that you should recognize, the integrase. So this indicates that QUK can form lysogens. So we went in the lab and we were able to isolate QUK lysogens. Down here, we see, again, some genes that could be early genes, DNA helicase, an exonuclease. This gene here is like a recombinase helicase. Genes that might be needed for DNA replication, and we don't know what this guy does right here, GP78. Some more genes probably involved in DNA replication, polymerases, etc., resolvase. Down here, possibly more early genes, a DNA primase polymerase, and then again, what looks like an operon of genes, and we have no idea what these genes do. So what stands out is the fact that we have an integrase, okay? An integrase indicates it can form lysogens. We know we've isolated these stable lysogens. The problem is we couldn't find a repressor in this genome. If you go back, you can see that these are all forward genes. So there's no region of divergent transcription, which often indicates, a leftward repressor and a rightward CRO gene. We don't see anything like that. And there's no gene called as a DNA binding domain protein. So nothing sticks out as the repressor. So which gene encodes the repressor? Well, we can begin to try to answer this question by asking, in fact, a different question. Given that the repressor should be highly expressed during lysogeny, we can ask the question, which QUK genes are expressed during lysogeny? That question we can answer using RNA-seq because this detects RNAs that are expressed during lysogeny or any other type of growth, and it will indicate which genes are being expressed at that particular growth phase. So here's what an experiment would look like if you were going to do RNA-seq. So we would have a flask of cells that would just be M. smegmatis. We'd have a second flask of cells that contain QUK lysogens of M. smegmatis, and we let these cells grow. And then we isolate RNA from each of these cells. And presumably, this would just be M. smegmatis, mRNAs that we're isolating. And down here, we would hope that we would be isolating M. smegmatis mRNAs, but also the mRNAs that the QUK lysogen is producing. And then what we do, essentially, is have those RNAs sequenced. So the RNAs are treated with DNase to get rid of genomic DNA and some other quality control steps. And then there's a library prepared because we have to turn the RNA into DNA so that the DNA can then be sequenced. And so all the little tiny RNA reads that are produced, remember these are going to be very short reads if you remember back to our sequencing unit, each of these reads is mapped to the cuke genome, and that will tell us which genes in this cuke flask were being expressed when we isolated RNA. But we can also map reads to the M. smegmatis genome, so we can even answer questions about which genes the bacterium is are being expressed during this lysogenic infection. While we didn't do this with cuke, another experiment you can do is take M. smegmatis and throw some lysate in there, and then at an early time point, maybe 30 minutes, isolate RNA, and then come back at a much later time point, at 150 minutes later, isolate RNA again, and then you'd be looking to see which genes are expressed during lytic infection, during early gene expression and during late gene expression. So again, another way to find out which genes are important for these different parts of the lytic cycle. We're just going to talk about genes that are expressed during lysogeny. So here's a look at a whole bunch of, each one of these little colored stripes represents RNA reads that were sequenced from the lysogen flask. And they have been mapped to this blue stripe showing a small portion of the cuke genome. So we have some software packages that will map all the reads to the sequence of the genome to which it matches. Now, this yellow bar is showing the very end of a gene, GP74. This region is an intergenic region. And then we see this yellow bar again is the beginning of what is GP75. And I've shown up here a little famirator cutout of GP74 and 75 of the cuke genome. And this yellow box represents the zoomed in region that we're looking at. Now up here we see a little graph showing the number of reads that map to each part of this genome. And so if you look over here at GP74, you can tell that there aren't very many reads that are mapping to this part of the genome. But as we move down the genome, just upstream of GP75, we see an increase in the number of reads that are mapping to this genome. And again, over the span of the ORF, we see an increase in the number of reads that are mapping to this part of the genome. So let's look at this a little bit zoomed out so that you can better appreciate the pattern of gene expression that's occurring. This is the entire cuke genome running across your screen. Again, up here, we don't see the reads below because that was super messy, but we can see up here the coverage graph that shows us the number of reads that map to each of these parts of the genome. Here is GP75 that we were just looking at. You can see how highly expressed GP75 is during lysogeny, whereas the genes surrounding it don't appear to be expressed during lysogeny. Over here, we see that the integrase is expressed during lysogeny, but probably the second most highly expressed gene during lysogeny is GP78. Now we know what the integrase does, so it makes sense that this is expressed during lysogeny, but we have no idea what GP75 and 78 do. There's no function called for these genes. So we had to go back and use some of our gene function analysis skills that you guys have been using in class. Another thing to note is GP75, which I'll tell you, had very poor hits on HH-pred. It looked like it had something to do with RNA binding and methyltransferase. Whatever it is, it's highly expressed. So we're super curious about this, but we don't think it's the repressor. And one reason is because there's 8 members of its fam, and we expect the repressor to be specific to cluster AC. Down here, looking at GP78, there's only 2 members in this fam. This is the second most highly expressed gene, and it's found only in cluster AC genomes. So what we can do is look at the possible function of GP78, and it had horrible HH-pred results as well. But it did, you can see that just by the blue lines, that there's pretty low probability and bad e-values. But the majority of these alignments had something to do with matches to transcriptional regulators. So although they were poor matches, the fact that the majority of them had something to do with binding DNA, and the fact that there's only 2 members of this fam, we're hypothesizing that this is probably the repressor for QUIC. So now we can dream up new bench experiments for both our GP75 and GP78. We can clone them into an expression plasmid. We can take that expression plasmid and pop it into mSMEG. And what that allows us to do is turn on this promoter and the gene downstream of it. And so now we have GP78 being expressed inside of an mSMEG cell, even though it's not infected with QUIC. We can make just GP78 turned on. And then if this is a repressor, then we would expect cells expressing a repressor to have superinfection immunity. So we can test this to see if these cells are susceptible to infection by QUIC phases. So this is just one experiment that we've cooked up using RNA-seq with QUIC. What questions can you ask about genes in your phage genomes, and how could you use RNA-seq to answer those questions?